Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, we have a few announcements to make. First, we'd like to thank our Dr. GPCR Ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Last month, we launched the Dr. GPCR Symposia event series. Our first symposium was on March 24th, 2023, and it was on challenging GPCRs. It was a day filled with amazing talks, great discussions, and we hope the beginning of new collaborations. In case you missed it, you can watch the talks in the Dr. GPCR ecosystem with your premium membership. Also, mark your calendars for the next symposium coming up on May 19th, during which we will hear from speakers on GPCR activation and signaling. At all our symposia events, including the one coming in May, all trainees are welcome to present a poster. There won't be any poster selections, and everyone is welcome to join us for the poster/networking time. For more information and an updated schedule can be found in the ecosystem. The easiest way to get to it is to use the links in the footer and look for Dr. GPCR Symposia. You can join us live by marking your calendar and becoming a Dr. GPCR Ecosystem free site member. To navigate the ecosystem again, please use the direct links in the footer. And now let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm very excited to have with me Chloe Hicks. She has accepted very quickly to join me on the podcast, and it's a topic Chloe and I are gonna talk about a little bit. But hi, Chloe. Nice to have you on.
Hi, Yamina. Thanks so much for having me. I'm honored to be here.
Thank you so much for joining us. And why don't we start by a short introduction, if you can tell us who you are and where you come from.
Sure. My name is Chloe Hicks. I'm a senior undergraduate here at Duke University. I grew up in Burlington, North Carolina, so I've been in North Carolina all of my life. And in terms of just how I got into science during my undergraduate career, I kind of had a few detours on the way before coming into the Rajagopal lab, where starting my freshman year, I joined a cancer lab. Couldn't quite tell you what the research was exactly because I wasn't very studious as a freshman, but that experience, I just didn't have it had a lack of structure that I needed of somebody telling me to come in and what experiments I'll be doing and explaining like the background to me. So like with that, there was only a semester of that before I started realizing maybe that wasn't for me. And then my freshman spring semester, COVID hit. So we all went home. And then the following semester, they have at Duke this thing called the Muser app, where you can kind of apply to research opportunities through that, which is a really great resource for undergraduates. But I joined a research project of a grad student that was looking to isolate a specific type of bacteria strand. And that whole project, if you could call it that, I was one step in an isolation protocol where I pipetted out the organic layer of a isolated or a filtration, and it just wasn't fulfilling at all because I was just pipetting a few hours every week. And fortunately, at that time, I started to get more into pharmacology and the idea of like the intersection. I like the intersection of biology and chemistry with a more of a medicine base than just a straight like biochemistry major. So when I was looking at labs that had pertained to pharmacology, Sudar's lab showed up. So I was reached out to him over email, and I was very fortunate that at that time, my grad student mentor Dylan was looking to take on another undergraduate student. So that's kind of my detour to getting into GPCRs. There was a little bit of trial and error, a little bit of luck, but yeah, that's kind of how I got into GPCR research.
That's awesome. And you mentioned Dylan. You're thinking about Dylan Iger, who was also on the podcast, and I loved talking to him. And since the whole lab works on CXCR3, which is one of my favorites, the Sudar's lab has a special place in my heart when it comes to the target itself. And we were joking earlier this week, or actually last week at the GPCR's Targeted Drug Discovery Summit. We were having lunch, and as we were talking about CXCR3, and Suda was like, yeah, you scooped us, Yamina. Thank you. But it's
Yeah.
interesting that as I told him, I didn't even know at the time that you guys were interested in CXCR3. So I'm just happy that I was able to put in a grain of sand into the CXCR3 realm.
Yeah, and I'm so thrilled you understand the struggle of looking at endogenous ligands with all the chemokine receptors where they just activate everything. But yeah,
Yeah,
it really is a great system for looking at for us by signaling. But
yeah, yeah. And it's funny because of the time when I worked on characterizing the different splice variants, and that was such a it was a hard project. It was during my postdoc, and I'm glad, and as Tom Sackmar said last week, you should be proud because there hasn't been a paper that came out with a single author in addition to the PI on it. And it was super hard, but it was so much fun working on this receptor.
yeah, and I'm glad you did too. So we didn't have
Yes, I kind of like paved the way for a lot of things and told you do not work on the alternative variant because that's a weird one.
yeah.
Although I'd be very interested in seeing that receptor or that protein from a heterodimerization perspective.
That would be really interesting.
But again, I don't work in the lab anymore. Maybe someone listening to it, Sudar, if you're listening and you feel like
Please,
taking
one more project. Yeah.
yeah. So tell me a little bit, you mentioned, being an undergrad and also starting to work a little bit in the lab and being like, this is really boring because I'm here pipetting a couple of times a week. But as a teenager or as a child, were you always interested in science?
So, as a child, I was quite delusional in my aspirations, where I think there was one point that sticks out to me where I wanted to be a zookeeper. And the reason for that is because the movie Zookeeper with Kevin James just came out, where he
Yeah,
talks to animals in it. So I
yeah.
just thought that would be incredible if I could do that as a career. So as a child, it was a lot of just fantastical ideas, but I never really or I didn't really consider science seriously until high school, where I realized I was good at science. So it was something that I liked doing. But in terms of just considering it as a career point or as medicine specifically, I don't think it was until I think my senior year of high school where my grandmother, she was diagnosed with lung cancer. And a part of that, yeah, well, a part of that coping mechanism for me was I picked up a book on immunotherapy. Specifically, it talked about chimeric antigen receptor T cells. I knew nothing about immunotherapy or cancer treatment at that time. So it was an easy book to understand in that way, but it was just so interesting to see the science involved in like being able to take someone's T cells and modify it so that it could potentially target an antigen in the body. I just thought that was so cool. But more than that, I just really was inspired by the hope of like the voice of it, which I've seen reflected through like so many different scientists that I've talked to since then, where they're just so optimistic about the potential of their fields and of the fact that they don't have answers yet, but that they're getting closer to it. And I just really loved that enthusiasm and that optimism for increasing the potential or just using that potential to further a field. So I think that's when I really started to consider medicine as well as science in a more serious career choice, I guess.
Yeah. So you mentioned science in general. Was there any specific topic that you preferred, or you liked all of them the same?
Well, in high school, it was chemistry because it made sense in high school. They make it seem like the pattern of chemistry is very simple. Then I got to college and I realized that's not the case, and I don't like chemistry so much anymore. So then I started getting, was more interested in biology, and originally was on the track for just like strictly biology. But I really did like the intersection with pharmacology of both chemistry and biology, where it was like you had more chemistry, but it wasn't like serious, like a high orbital, like all of the different theoretical stuff of chemistry that kind of went over my head a little bit and I wasn't as interested in. But yeah, I think just like the more pharmacological basis behind science is really what interests me, like different sort of targets or therapeutics was something that I took a lot of interest in.
I love it, and I can relate to the chemistry thing. I loved chemistry. up until I got to college and university, and I was like, well, despite my best effort, this is just not working. I just don't get it. And I even deferred one course to the summer because I was like, I just can't handle 6 or 8 classes, and chemistry was one of those where I was like, okay, I just hope to pass.
Exactly. Yeah. Organic chemistry was the same way for me. I was like,
yeah.
yeah.
And I really wanted to understand. It's funny because we had 2 exams, one mid-semester, and then at the end, the one during mid-semester was always got 80-85%, and at the end tanked it, but thank God it kind of leveled out. But it was just such a difficult, I just couldn't get it.
Yeah.
So, but then I do understand that the chemistry must be respected.
Exactly. We need it, but yeah.
No, agreed.
Interesting part to me.
Yeah. And so you also mentioned this, so now you work in Sudar's lab, and you're a senior undergraduate.
Mm-hmm.
What does that look like? What do your days look like?
So at this point, as a senior, I'm taking 3 classes currently, but they're a lot more low yield than they were my sophomore and junior year, where I was trying to finish up the pre-med prerequisites like physics and biochemistry and organic chemistry. So my days now are just very much I'm in the lab when I can be, which is most of the time. So I'm currently in the process of working on my senior thesis right now, which is making headway, but we still have links to go. But yeah, so in terms of like the layout of what my undergraduate experience in the lab looked like, it was a few hours a week in my sophomore year where I was starting to get comfortable with the assays. I stayed for the summer and got a lot more comfortable with those assays and understanding the GPCR field in general. And then junior year, it kind of amped up. Like I was in lab between classes, but it wasn't to the extent that it is now, where I work primarily independently. Dylan's now in the hospital, and I work alongside Julia. We are both seniors that are just in the lab when we can be, which is most of the time, and just working on getting this project out. So it's really fun time. I really love the independence that I have this year, especially, and I'm just enjoying it and learning from all of the great grad students in Sudar and everyone in the lab. It's a good opportunity.
I love it. I want to point out that I'm very happy that you accepted to join on the podcast, and we've talked about it a little bit before hitting record, because we've been struggling to get trainees on the podcast, and I've been emphasizing this, and that's why I'm taking a minute here to emphasize to listeners that everyone is welcome in the podcast. You do not have to have 50 years of experience in the GPCR field, because your experience is unique and it might help someone listening to your experience. Plus, also as a guest, I feel like it's good, wherever you go, you say, well, I was on this podcast. And it also helps. It also helps. No worries. No. There's a delay, so if you feel like I'm interrupting you, I'm sorry about that. But I think it's also a good thing to put on your resume at some point, or your CV, because you also develop communication skills and soft skills, and you can you show that you can have a high level conversation around a scientific topic.
Yep.
So this is where I'm going to conclude my invitation to everyone in the GPCR field. If you received an email from us, please join us on the podcast. If you have not and you want to join us on the podcast, there is a submission form on the podcast page at the top where you can just apply. What it means is that you just tell us that you're interested, and we're definitely gonna reach out. Getting back to you, Chloe, it's funny because you were mentioning, between courses being in the lab. I did exactly the same thing. I had a summer during undergrad where you had to work in the lab. And after that, instead of going to do any random job, I asked my PI, I said, look, I can come in between classes. Is there a project for me? And I ended up doing a master's and a PhD and spending almost 10 years in the same lab because it was so much fun. doing this type of work. What was your experience when you joined Sudar's lab, and like, what did you like about the lab, about the projects, about the people that made you stay, basically?
Yeah, well, I think one of the big things at the start of it that really stuck out to me is that I would attend the lab meetings, and everyone, like, people would present data, but throughout it, all of the grad students were participating and asking questions and bouncing ideas off of each other. So, and it's still that way with a lot of the lab meetings where it's partially you're presenting to the data, but it's also just everyone's kind of bouncing ideas off of each other, bringing up other previous papers that cited this, and it's a very creative environment for that. It's very it's very, what is it, collaborative in that way. And I think that's replicated in a lot of the GPCR fields or the GPCR labs here at Duke. It's an extremely collaborative place I've found, which I personally really appreciate, where you can go to other labs for and questions about specific assays that they do or reagents. And we have data club, which is this thing where once a week people present their data to all of the GPCR labs here. And it's just an extremely collaborative environment that I really appreciate, having just the insight of all these brilliant scientists. I really appreciated that. And then the other thing was just I had the mentor Dylan, who invested time and energy into like teaching me the field, asking me questions about interpreting data, like challenging me to read the literature and to understand further what we're doing, to the point that I could then figure out the next step in that process. So I really just appreciated all of the different like collaboration, creativity, independence, and challenging environment that I was given through Sudar's lab.
That's amazing. And as I mentioned, I've interacted with Dylan. He was on the podcast. He also presented at the Dr. GPCR Summit. And oh my God, his presentation was like so amazing, spot on. He's amazing.
Yeah.
And I'm sure he's going to do amazing as an MD PhD as well. You mentioned a little bit about how you got into studying GPCRs, and how you well, actually through Dylan, basically, and through being in Sudar's lab. But do you remember the first time you heard about GPCRs?
I think it was looking at Sudar's page, because at that point I hadn't taken biochemistry, and biology intro classes were mostly DNA, clone, or just DNA based in evolution. So it was just, I looked at his page and I said, what is a GPCR? Apparently it's important because it's everywhere in the body, and there's a lot of the receptors. But yeah, that was my first exposure to GPCRs, was through this lab and learning about the different mechanisms. Because when I was studying for the MCAT, they would have like the review of GPCRs, and it would just be, they activate G proteins and it signals, and that's all they do. And it's just so interesting to see the difference between kind of the literature of how people teach GPCRs versus the ongoing science behind it, and how much more nuanced it is when you look at it through not a class that's trying to simplify it down outlook.
Yeah, it's just one topic among many, and when you work in a GPCR lab or you're exposed to it, you're like, okay, wait a minute, this is way more complicated than we think.
Yeah, the G protein isn't just activating cyclic AMP. That's not
right. And it's heterotrimeric. It's just not one thing. But that's great. I'm gonna ask the question. I kind of know what the answer is. What's your favorite GPCR?
Well, I don't know. I might throw you for a curveball, because I know yours and everyone here, or in Sudar's lab, is usually CXCR3, but I'm very selfish in that my senior project has been mostly on the atypical chemokine receptor, ACKR3. So I'm going to have to go with that one. The reason for that is just because, and this is part of the project, what we're looking at now is it's a really good system for looking at non-canonical signaling effectors of GPCR signaling. Because for those listening who aren't as aware of ACKR3's function, it's also known as CXCR7. So if that's confusing, apologies. But so it was originally known to be a scavenger receptor, where it was thought that it would just uptake CXCL12 and isolate it from CXCR4 to stop it from activating. But what Sudar himself found out was that ACKR3 is able to actually signal through beta-arrestin. It just doesn't couple to G protein. So because of that, we consider it a beta-arrestin biased receptor. So then another paper by, I believe it was Nicholas Montpass, came out where he showed that when you do a mutation that inhibits beta-arrestin recruitment to the receptor, you still get internalization of it and you still get signaling output of ACKR3. And then I believe it was like a year later, Frederick Saber showed that you need phosphorylation of ACKR3, but you don't need beta-arrestin recruitment in order to get a signaling output. For them, they were looking at neuronal migration, but that just indicated to us that this receptor is able to signal even without beta-arrestin, even without G protein. So there's potential for, there has to be another protein effector maybe that's playing a role in having this receptor signal. So it's just, in my opinion, it's a great model for looking at how a receptor can signal without beta-arrestin and G protein, because I think the binary model we have where they can either signal through G protein or beta-arrestin is just limited in what is actually physiologically possible for a receptor. I think they found a lot of different ways to be able to signal despite our best efforts of knocking out beta-arrestin or G protein. So it's a really interesting, and I'm excited to pursue it more. But yeah, I think ACKR3 I would have to go with. So apologies. That's
no, I love it. And I formerly known it as CXCR7 as well. And I was trying to think about, so to make a long story short, Nicolas Mompa was a graduate student in Nicolas Evequera's lab where I did my PhD as well. And I know Nicolas very well. And the first paper that came out from our lab, and I think it came out around the same time as Sudar's paper. So that's why I'm like, okay, which one? And so basically, long story short, it was the first author was Kalatskaya Irina, and I was co-first with her. And that's the paper where we were looking at beta-arrestin recruitment to CXCR7. And we identified AMD 3100 as a positive allosteric modulator of CXCR7. And I remember that it was a struggle because the beta-arrestin recruitment, I think we had CXCR4 as a control, and CXCR4 wasn't as potent, and it was just this whole project, but it was a really cool one. And I think it was a collaboration with industry. So CXCR7 also has this special place. And at the time, there were all these chemocentrics. Tom Schall's group at Chemocentrics was very interested in CXCR7 as well. So, but I think it's a really cool receptor, and it makes you wonder, what does it do in the body in general? And then I'll take it one step further, since it also shares a ligand with CXCR3, because I always have to bring it back to CXCR3. I wonder if CXCR3 and CXCR7 form heterodimers. I don't think anyone looked at it.
Yeah, that's an interesting, I have no literature on CXCR4, CXCR7. I don't know if I've necessarily seen anything with CXCR3. I wouldn't doubt it if it did. Yeah. I think it's a lot less explored if it's so.
Yeah, 4 and 7 heterodimerize. It's a paper that came out maybe a year before I joined Tom Sackmar's lab, and they were working on CXCR4 and 7. So when I joined the lab for my postdoc, I was like, okay, has anyone ever looked at 3 and 7?
Yeah.
And that's when Tom said, oh, wait a minute, 3 has different splice variants. Irina, do you know what these do? And that was the beginning of a 5-year project to figure out what they do.
Another project, just to have that on too. But exactly. Yeah. And I think like the point you made up about the fact that understanding the function of this receptor is well taken, because we really don't understand much about the cellular output. It's known to be overexpressed in cancer cells, it's expressed in T cells, and it's involved in inflammatory pathways. But I think a lot of our understanding of ACKR3 is still just limited to this idea of it as a scavenger receptor. I think an example of this, I was just reading yesterday, it just got published yesterday, but this lab, this Maria Steele is the first author of it, but it's looking at the tumor microenvironment of T cells, and they saw that you get a flip of expression in CXCR4 and CXCR7. So with CXCR7, otherwise ACKR3, it's upregulated in these tumor environments, whereas CXCR4 is downregulated. So their hypothesis with this paper was that you get this downregulation because CXCR4 is involved in tumor egress. So their idea was that maybe CXCR7 is being overexpressed to scavenge this CXCL12 and stop CXCR4 from activating. But I feel like that's such an energetically like a very you need a lot of energy to do that, in the sense that there are easier ways that the cells have figured out, I feel, to cease one receptor from activating that doesn't require a whole other receptor being overexpressed. So I think just this simplicity of just seeing ACKR3 as just there to stop CXCR4 signaling is just a bit limited, in that it's probably doing something else, some other function, but we just are very limited in our knowledge of it right now. So I agree that that's definitely something that needs to be explored further.
No, agreed. And you made a great point about our tendency as a field to look at G protein versus beta arrestin. And I don't think it's because we don't know any better. I think it's because those are the two pathways for which we have the most tools.
Yes.
And we're all aware that people are going to email me like, how dare you say this about all of us? But at least and let's bring it down to me. We know that this works, and you need to better understand what GPCRs do. So you use the tools that you have, but there is this subculture, from what I've seen in the literature, where people are trying to understand, okay, what else?
Yeah.
And what else can we be measuring, and can we go further downstream? And I think the holistic view is trying to measure everything, but then we cannot yet measure everything. And what do you do with the amount of data that generates? So what does that mean?
Yeah. Also, just how to interpret that data with like something like ACKR3 with you when you have the chemokines that like are activating other receptors, and you can't know for sure if this signaling output's because it's activating CXCR4 or CXCR7 or CXCR3 if you're looking at CXCR11. So
exactly, which is why I love the chemokine system. I think it's a funky one. It's
funky little thing. Yeah.
We still, I feel like it's the highways in LA, that they show on TV, and then one goes
going in the right direction. Just
they know what they're doing. We don't yet.
Yes, exactly.
So I was wondering, I'm trying to formulate the question here, and since we're talking about methodologies and better understanding how the chemokine receptor system works, if you had a magic wand and you could, make an assay a reality to better understand your system of interest, what would that assay look like? Or what would you do with that magic wand at that point?
So I don't know if it's so much a magic wand, in that I just want, when we talk about the chemokines, just if there's a way to modulate so that they only activate one receptor would be really cool for me. But also with our lab, we have a specific interest in what we call location bias, which is this understanding that the receptor can signal through different subcellular compartments in the cell, and we show that with CXCR3, but we don't really understand why, like, the signaling profile of this receptor changes in these environments. Is it because of the system? Is there more G protein available in these subcellular compartments than beta arrestin? Is it because the pH is different, or is it because the conformation of the receptor is different in the endosome? So I think it would be interesting to get, I know people are working on it now, cell permeable nanobodies that can target or put these or receptors in specific conformations, or not even put them in specific, but detect conformations so that we can see, does the receptor look different? Is there a conformational change in the endosome that's what's regulating this difference in signaling profile, or is it something about the environment? I think just having that tool and having that knowledge of how this receptor's structure is different at different areas of the cell could really help us further understand why the signaling profile is just so different depending on where you are in the cell.
Ooh, I love it. And you were talking, I was like, yeah, we could look at lipid composition as
Yeah,
well in these different
exactly. Yeah.
compartments. And then we could, oh, well, we could, it would be really great to be able to sample the conformational landscape depending on where you are
Yes. Yeah.
in the cell. I think it's brilliant. It was really, I think we still have some way to go until we get there, but it would be really, really interesting to be able to compare based on the location of the receptor, what changes in its conformation, how do lipids influence that? Are there any other interactor proteins in those compartments that influence receptor function
Yeah.
to better understand what happened? Why would you signal
Mhm.
in an endosome?
Yeah. And it's probably a combination of all of these possibilities plus more. We
Yeah,
don't even have the tools to even
yeah,
figure out at this point yet,
yeah.
but it would be interesting for sure.
But already knowing these smaller data points would help to better understand. And also I'll take it to the next level where I'll say, can we link any pathophysiological state to some conformation in some subcellular compartment and say, well, this is the route that we want to target, and
Mhm.
come up with either small molecules or antibodies that are obviously getting to the right spot that would modulate this.
That's the dream right there.
Yeah, I think we're just speculating here, but that sounds like a really cool topic to think about and hopefully get more information about as technologies develop.
I agree.
I love it. I love it. So there is enough work for a lot of graduate students and a lot of
5 other
PhD.
projects, yeah.
Yeah.
If anyone wants to take them on,
Postdocs. We won't be out of projects on GPCRs for a while.
certainly.
So the other question I had for you, Chloe, you're once you graduate, you mentioned, being in the dual MD PhD program. Is that the route that you're considering taking?
Yeah, so initially when I joined the lab, I was very much MD focused based, and I just wanted to look at research, as an interesting thing to do on the side and help with applications for medical school. But just the experience in this lab has just been really great, and understanding the research and how interesting and how much there's left to learn. It really inspired me to consider MD PhD. So yes, on the currently on I'm focused on applying to MSTP programs in May of the next year. So I'll have like one gap year in between where I'm going to be working at the NIH. So hopefully it's not going to be on GPCR stuff. Unfortunately, I'll be leaving the field just for a little bit. But yeah, I'm really excited about the prospect of applying and talking to people about my research and hearing about theirs to a greater extent than I've done so far.
That's great. Can I ask where at NIH?
Yeah, I'm joining Ian Fraser's lab. He
Mhm.
works with macrophage, understanding just both like the genetics and the signaling mechanisms of these different macrophage pathways. So I'm really excited to learn more about that field. I definitely need to do my homework on all of that signaling because it's so different from GPCRs.
Yeah.
But yeah, I'm excited. Yeah.
Well, you'll get to encounter GPCRs
Oh,
and macrophages for sure. So
they're
you're
still there.
you're not. I wouldn't say that you're leaving the field. You're just readjusting your view of the field for a while before coming back
Yeah,
to
they
it.
never go away. They're always there.
Definitely not. And what institute is Ian Fraser's lab at?
It's a great question. I should know the institute in the NIH. It's in
NIAID by any chance?
yes. I'm sorry. Yes, NIAID. I'm sorry. I
Okay.
need
No,
to.
I mean, there is the reason I was asking is because the name sounds familiar. And
Mhm.
I also spent 3 years in the B cell molecular immunology lab at NIH, at NIAID. And initially I wasn't supposed to work directly on GPCRs. It was more mouse work, but honestly, I had such a hard time sacrificing the mice that I came up with a really cool Brett project on this very interesting chemokine receptor and, built out an entire project which complemented really well a mouse project, and we have mouse data that goes with it. And the name sounded familiar, and The other thing is that NIH, there's all these institutes, and I think at some point somebody said we need to stop naming the institutes, and we need to stop having as many. And you'll see once you get to NIH, there's all these acronyms that are very NIH specific.
Mhm.
When you're like, okay, I need a glossary here, and then write this down so that I can learn it. But you'll learn it pretty quickly.
Yeah, I just need a dictionary of all the different acronyms to start
Yes,
learning.
exactly.
Yeah.
Exactly. And I think you're going to be on the main campus in Bethesda, which
Yeah.
is a huge, huge and beautiful campus. I think you're gonna love it.
I'm excited. I haven't been outside of North Carolina, so DC is going to be an interesting, exciting change for me, and just in terms of environment. So I'm not
Yeah.
stuck with the extreme temperature changes of North Carolina day-to-day basis.
That's amazing. Since you're getting into a new lab soon, let's if you have a couple of minutes, let's have a chat around how did you pick the lab and what was important to you when you picked Ian Fraser's lab?
Yeah, so I guess I'm in the minority in terms of NIH uh, post-bac applicants, where I really just wanted to do 1 year. And my reasoning for that is just MD PhD is about like 8 years of my life, and I feel prepared and developed enough as a student and as an applicant now to just go ahead and apply, and then just have that year to just immerse myself into a completely different field and learn the different assays and strategies of that field and the literature, and hopefully be able to bring my own insight and experience from this lab to maybe help contribute other possibilities of assays or experiments that could help. But basically, I just really was invested in the people, like just having a PI that would be
Yeah.
very first of all, understanding of time commitments, but also willing to teach me and excited to teach someone about the field. And both him and the grad student Clinton, who I will be working under, just seemed very enthusiastic about mentorship. And mentorship has always been something that's really important to me. So
Yeah.
just having somebody who has the same values of what it takes to be a mentor is really important to me.
Clinton
So
Bradfield, by any chance?
yes, that's
Oh my
him.
God, I know him.
It's such
He's
a small
awesome.
world. He is.
He
Yeah.
is awesome. He is really, really a nice guy. And we actually collaborated. I don't know what happened to that data, but Clinton is just phenomenal. I think you're gonna
I'm
love working
so
with
glad
him.
I have your recommendation as well. It makes me
That's
feel
good. That's no, happy. And please say hi to him. And I'm gonna shoot him an email after this. You said Clinton. I was like, wait a minute.
yeah.
Wait a minute.
Small
And
world.
it's so funny because the Ian Fraser's name sounded so familiar, and now I know where their lab is, and
Yeah.
we've collaborated to the extent that Clinton is an amazing scientist, and he helped me with creating CRISPR cell lines, and so we've made GRK2 CRISPR cell lines, and it's a long story, but I was working on S1P1 receptors, and there's 5 of them, and we needed S1P1R because the Hicks cells would respond to the ligands without transfecting with pcDNA transfected cells. So I knocked out, or at least I tried to knock out all S1P receptors from the Hicks cells so that we can have a clear background and then retransfect the
Yeah.
receptor. And I knew nothing about CRISPR and how to do this. And Clinton was such an amazing support in that regard. He was like, "No, this is what you order. This is what you do." And I know he was just. I'm glad you're gonna be working with him.
Yeah. I'm glad you know him and can vet for
Yeah,
his.
totally.
Yeah.
I think he's really fantastic. Awesome. And what next after NIH? What does that
So
look
that
like?
will just be applying for MD PhD programs, MSTP. I don't have a particular school or location in mind. I think it's just gonna be just gonna base it off of just my experiences with
Yeah.
interviewing and everything. So I don't have as much of a clear outline after NIH, but I know I want to continue pursuing research and medicine simultaneously. So I'm excited about the prospect of MD PhD. So
That's cool. And it's funny because I feel like, if I remember correctly, Sudar is also an MD PhD.
Yes, I believe oh my gosh, he started college so much younger
He's
too.
16, I
And
think, or something.
yeah, yeah, exactly.
Yeah. Yeah.
No gap year. It's just straight through. Insane, insane.
I know. I think he's in that unique category of people. I mean, 16, come on. It's
Super
amazing.
smart. Oh my gosh, yeah.
And his brother as well. So, if
Yeah,
I
he did
understand
like
correctly.
13 or something. He did college or
Yeah.
yeah, just
Wow.
overachievers,
That's a
truly.
whole other level of smartness here. I feel
It's just like slackers for taking time.
exactly. Exactly. No, that's amazing. And I think a lot of you in the lab are um thinking about this profile of MD PhD. And I feel like there's that connection that you form with Sudar, and then you're looking at his experience. You're like, oh, actually, I really like this. And I have to say that, well, I've interacted now with you, and you've presented at the Dr. GPCR Summit, and I had Dylan in the same, you know, presenting at the summit, coming on the podcast, and later this week we're gonna have somebody else from your lab. I'm not gonna say who because the order, well, her episode is coming after yours anyway, so it's the right order. But I feel like all of you are so articulate, and I wish I were that articulate at the time where I was an undergraduate. And I think it's kudos to all of you guys. I think you're all amazing.
Thank you. I really appreciate that. And yeah, I agree with you. There's a lot of MD PhDs that have come out of the lab. I know there's two other postbacks at the NIH currently right now that were also mentees of Dylan's, Noe and Cole, who are also in the process of applying to MSTPs. And I just think it's just really, this lab has been just a great opportunity to see that. And I feel that a lot of times you're limited on resources telling you about MD PhD if you're not in a lab or know somebody who's doing MD PhD. You just have a lot less access to resources as in comparison to MD resources or people telling you what you need for MD. There's not as much exposure to the idea of being a physician scientist, which I think is a shame because there's a lot of super intelligent and creative people that would, I feel, be great at this opportunity. But yeah, I think just expanding the resources available and just the knowledge of what a career as a physician scientist looks like might recruit even more undergraduates to start considering the possibility that it might be something they really find a lot of worthwhile in. Yeah.
What kind of resources are you thinking? I'm trying to think because I agree. I didn't know about the lack of resources around, the MD PhD track. I do know more about the lack of resources around the non-academic tracks, because that's also a black hole for a lot of trainees. But what kind of resources are you thinking of?
Yeah, I mean, when I say resource, it's very broad, but what comes to mind for me, Duke here has so many great resources and opportunity for understanding for their pre-meds and people who want to get into the medical school, and they're really, really on top of that, like setting up from freshman year, like what you can look for that. But a lot of it I've noticed through preparing for applications, a lot of it's just MD based, and then it's kind of second, like you have to go out of your way and ask about MD PhD or about DO or any of the other combination resources to really get an understanding of how it's different. So like a lot of their preparation, for instance, they when they're telling you what you should have on your application, research is the bottom of the list. They're like, oh, it's nice if you have it, but you don't necessarily need it. And a lot of just the info you're getting is just very, very MD centered, where for like MD PhD, research is obviously a lot more important. So I think not only that, but also just exposure to people, like having people that you can come in contact with, because there's so much of a smaller proportion of MD PhD students in comparison to MD. You really have to go out of your way or know somebody who knows somebody who's an MD PhD if you want to like ask somebody or to get into a conversation about what that entails, or you can just get really lucky like me and join a lab where there's a lot of MD PhD resources and opportunities and people to ask about their experience. But I just feel like I got very lucky in that, and I think we could be doing more in terms of increasing access to people and to information on PhD and how it's different from MD, because it's a different process and there's other things to consider that are.
That's good to know. We'll keep that in mind, not necessarily in the context of Dr. GPCR, but we're working on, it's a long story. It was a project that we started that got kind of stopped, and we're trying to get it back on track. And I'm always interested in hearing more about what do people need. Mm-hmm. Like, you're a graduate student or you're an undergrad and you're planning your career. What is the information that you're looking for? Is it available? And if yes, where? And is it the complete information that you're looking for or not? And that's really valuable. So thanks for that. Sure. Right. No, that's really cool. Now I'm trying to think, like my brain started working. We're going to have to talk about it once we start recording. All right, last but not least, top 3 aha moments that you had.
I feel like at this point in my career, it's a lot more, I don't have as many aha moments more than just, oh
no.
Not oh no, but just like, okay. Oh, I was doing this completely wrong. But I think the situation that stands out for me was just this summer, I was really working on cloning a nanobit construct for the ACKR3 so that we could look at potential proteins recruitment to the receptor of like non-canonical signaling effectors. So while I was cloning this, I was doing restriction digest, and I just left out, like it wasn't working for months, and I just kept doing it, and I was getting really frustrated. And the result was I wasn't putting an AT at the end of my primers to like allow the restriction enzyme to have room to actually recognize the enzyme binding site. So once I changed the primers, it worked like in the first time I did it. So it was just more like yeah, exactly. FOMO. It wasn't really aha. I didn't reinvent the wheel. I just figured out, oh, I need to read protocols more closely. But I think in terms of aha moments, I don't really have specific ones, but I think I just want to replicate the idea. I feel one of your podcasts guest said, I think it was Michelle Bouvier, said like the excitement of just seeing data for the first time, like being the first person to see that data and being able to interpret it without the bias of like whatever anyone else is saying their opinions on, and just having everyone just having to interpret that and what that means. The next step is just something that I always enjoy doing. So just the aha moment of seeing whether your assay worked or you're seeing something with significant difference is always just really, really cool to me in that sense.
It's rewarding for sure. You get what I call that dopamine rush.
Yeah, exactly.
We all long for.
Yes, it's working. I didn't completely right. Yeah.
Yeah. No, agreed. All right. Thanks, Chloe. I really appreciate your time.
Of course. Thank you so much for having me.
Of course, and I wish you the best of luck at NIH. I can't wait to email Clinton. I'm like, hey, you're getting a star here.
Yeah. Well, thank you so much. I really appreciate and I enjoyed talking to you.
Thank you so much.
Thank you.
Take care.
Bye.
Thank you for listening to this Dr. GPCR podcast episode. We would like to thank our guest, our Dr. GPCR team members, Attila, Ines, Monse, Ivana, Andrina, Balint, and Julia. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with us and with our partners directly in the ecosystem, so make sure that you join us today. Also, please subscribe to the Dr. GPCR newsletter. Find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial on our website. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. You can always email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.